Hi, my name is Sara Aghamiri. I'm a researcher at the University of Nebraska-Lincoln, and I'm going to present our poster titled The In Silico Experiments on Antigen-Presenting Cell Signaling Pathways. This poster presentation includes the introduction, methods, results, conclusions, and perspectives. Biological processes rely on complex, nonlinear network systems that span multiple layers of biological organization, ranging from molecular to cellular to organ and organismal levels. Developments in omics technologies revolutionized the research in immunology field, which significantly increased the demand of integrative approaches to address novel predictions and analysis. One of these approaches is computational modeling that provides executable dynamic networks revealing system-level behaviors through in silico perturbations. We aim to provide a multi-scale multicellular virtual immune system well-suited to exploring the immune system's fundamental properties and modeling immune responses to specific diseases. Antigen-presenting cells, or APCs, are immune cells mediating immune response by processing and presenting antigens to lymphocytes such as T cells. In this presentation, we will show you our mathematical model for dendritic cells, or DCs, which are considered critical APCs playing an essential role in immune responses. The process of curating the DC model started with the literature search to gather the available knowledge for APC signaling pathways. We built a Boolean network of DC that utilizes logical formalism, enabling in silico simulations. This model provides a platform for researchers to generate new or rank existing hypotheses before starting the wet lab experiments, saving time, energy, and materials. We curated the DC model in the Cell Collective, which is a modeling platform for building, simulating, and analyzing mathematical models. The DC model is a manual curated, entirely annotated, and expert-validated dynamic network. It contains more than 300 nodes and interactions presenting the APC pathways based on human cell-specific experimental publications. The model comprises different cellular compartments, from extracellular space to plasma membrane, cytoplasm, nucleus, and secreted molecules that facilitate tracking the flow of information in the system. The model also includes surface markers and contains detailed annotations that can address different DC subtypes. Here we can see the environment of performing in silico simulations in the Cell Collective platform. For example, we would like to know if we set an initial condition for the pathogen in extracellular space, what will happen to the secretion of a specific molecule. We can investigate the behavior of the DC under different environmental conditions or gene perturbations. The DC mathematical model can investigate several pathways at a time, while this may be a difficult task in wet lab experiments. We performed pathway enrichment analysis and found activation of multiple kinase signaling. This analysis emphasizes the most important pathways involved in DC biology, providing insight into the key signaling regulators driving APC activity. We also studied the most important functions related to the model components. The majority of the DC network is involved in the positive regulation of leukocytes, which is inconsistent with the definition of APC function. Besides, the model could segregate between the crosstalk of DC with different subtypes of T cells. To conclude, the DC mathematical model provides a platform for researchers to perform in silico simulations before starting the wet lab experiments. The investigation of the DC model shows the dynamic crosstalk between innate and adaptive immunity. The model also presents the regulation of canonical and non-canonical intracellular signaling related to APC function. Analysis of the DC model can guide future experiments for generating data in this field. The perspective of this work includes generating data to address the DC pathways for which there is a lack of knowledge, validating the DC model predictions for activating or inhibiting immune response, and constructing the multi-scale model of DC for the virtual immune system. This work is supported by NIH grant to Dr. Thomas Helikar.